So what right now I'm talking about, monosaccharide. So what monosaccharides are? Monosaccharides are the simplest carbohydrate. Why they are called simplest? Because they cannot be hydrolyzed further into some simpler unit, right? For example, glucose, fructose and many more. These are some of the molecules which you cannot hydrolyze them, into further smaller unit. But monosaccharide is the topic, and then I will talk to you about glucose. So monosaccharide I have mentioned here, and I'm going to, write a little bit about what monosaccharides are. These are simplest carbohydrate. These are simplest carbohydrates. So these are simplest carbohydrates. Now what I'm going to do, I'm going to classify it. So it has basically two types. So the one, the first one is called aldose. The first one is called aldose, while the second one is called ketose. Now what aldose means? It means, that it has polyhydroxy plus one aldehyde, and that is why it is called aldose. Al refers to aldehyde and OH refers to basically sugar. You need to understand that OH refers to sugar and Al refers to aldehyde. So this sugar is basically about, a sugar which has hydroxy group. Along with that you have, aldehyde group. Now if you want to know what are the example here, then I must take you the two common example I remember is glucose and fructose. So these are two very common examples, glucose and fructose. There are so many more, but I'm completing right here. Now let's talk about the ketose. What ketose are? Ketose are actually, these are also polyhydroxyaldehyde. Polyhydroxy with ketone. Polyhydroxy with ketone. Now if you want to know what are the example, then very simple example is that I have messed up here. The fructose is not, aldose, only the glucose is a aldose here. So, fructose is a ketose actually. So fructose is the example. Fructose is an example. Now what next we need to know is that, what this glucose is, because this is going to be the biggest part of this biomolecule chapter, where you have to study so much about glucose, right? So for that, what I'm going to do, I'm going to talk to you about the glucose. So glucose is the one, I'm talking. So the first thing I have in my mind is its sources. Sources of glucose. Sources of glucose. What does mean? That what are the things that we have around us from glucose can be extracted? So it has the first one, all the sweet fruits. These are present in all the sweet fruits. It is also present in honey, and its biggest source is ripened grapes. So grape is the largest source of glucose. Let's talk about the preparation of the glucose. The third very important thing I'm going to talk to you about, preparation. So preparation of glucose is the next topic. Preparation of glucose. How you can obtain. What? Glucose is one of the most abundant chemical on earth, as it is present as a constituent of cell wall, because the cell wall is made up of cellulose and cellulose is made up of glucose. So we can indirectly say that it is one of the most, abundant substance on earth, and basically it's coming from the, plant. And where the plant is, getting it from? Plant is getting it from photosynthesis. That is something you do know. So glucose is the, biggest source of glucose is the photosynthesis. So what I'm going to do, I'm going to make this fourth one right here, by photosynthesis, by photosynthesis in the green plants, green plants. Now we can talk about the preparation. The first preparatory method which I remember is, hydrolysis of sucrose. So it can be obtained by hydrolysis of sucrose. Hydrolysis of sucrose. Now what does hydrolysis involve? You have to take, sucrose C12H22O11, and you need to add water into it. You need to add water, and along with that you need small amount of, acid, because it acts as a catalyst there. And then this undergo hydrolysis, it undergo breaking, and this produce, glucose C6H12O6, and along with that it also produce C6H12O6. While this one is your glucose, while this one is an isomer of glucose called fructose. But fructose is not our topic. We are mainly concerned about the glucose. So the glucose first preparatory method involves hydrolysis of sucrose. Let's talk about the second one. What we can do, we can do hydrolysis of starch. Hydrolysis of starch. Now, this is the formula of starch here. This is your starch, and it has to be, it is a polymer of this molecule. So I'm placing N over here. And what you need to do hydrolysis. And for hydrolysis, you need to have small amount of acid. Along with that, 2 to 3 atm pressure, and around 343 Kelvin. These are not very important, but you should try to remember or recall everything. So at this particular temperature, when this starch, is mixed with water, I forgot to write this water, right? It produces, glucose. It produces glucose, C6H12O6, and it's definitely going to be in large numbers. So I'm placing N over here, and this N represents that one molecule of starch can produce a huge or a very large number of glucose. So what this is your starch here. You need to pay attention. This is the starch and this is your glucose. So these are the two preparatory methods you have in your book been described, right? And apart from that, I want you to concentrate about, whatever I just said. I let you know that monosaccharides are of two types. They are called aldose and ketose. What aldose are? Aldose are polyhydroxyaldehyde, and what ketose are? Polyhydroxyketone. The first example, the example for the first one is glucose, and for the other one is fructose. We then started with glucose, and what glucose, where it is coming from? Glucose is mainly coming from sweet fruits, honey, all the ripe grapes, and also by the photosynthesis by the green plants. How we can prepare glucose? By the hydrolysis of sucrose or by the hydrolysis of the starch. What starch is? C6H10O5, multiplied by N, because N represent that it is a polymer of this particular substance. So when you hydrolyze this starch, you will obtain N number of glucose. So these are some of the method of preparation of the glucose and things about. In the next lecture, I will talk to you about the structure of glucose.